This is part 7 of the 8 parts of genomics. And today I'll be talking about organelles primarily, and then get into the first part of eukarya. And then the final one, part 8, will be to finish up on eukaryotes. Materials for this part can be found in chapters 31 through 33 of the Integrated Molecular Evolution 2nd edition. I will first talk about organellar genomes, including mitochondrial and chloroplast. You can see both are, of course, obligate parasites, essentially. They're dependent on their hosts, and they originate from bacteria. So in this case, mitochondria from an alpha proteobacterium and chloroplast from cyanobacteria. Although the range of genomes for mitochondria is quite large, part of that is because in plants you have a large range of genomes, even though they still have a small number of genes. All organelles appear to have entered the host cell by a similar mechanism, in this case bacteria, but also there are organelles in protists that are red and green algal cells, so those are eukaryotic organelles. So in this case, you have the bacterium entering the host cell that does not have a mitochondrion. Then you get a time when they are mutualist, that is, they're gaining from one another. As they do this, the organellar genome tends to lose genes, and a lot of those are sent, often in multiple copies, to the nucleus, which soaks those up into its genome, incorporates those into the host genome. And so eventually you have many of the organellar genes being expressed from the nucleus rather than from the organelle. And then they have signal peptides that will carry those proteins into the organelle, where they enter whatever pathway they're involved in. Here's a diagram showing the ranges of various groups of organisms, organelles, and viruses. So you can see free-living organisms need a lot more DNA and more genes. And as some of those move into becoming parasites or organelles, they are reduced in size. And finally, you have obligate parasites and organelles that have lost more DNA and have moved further towards the left, or having smaller genomes and fewer numbers of genes. This shows that relationship in a slightly different way. So as you move from a parasitic or commensal relationship where the parasite tends to have smaller genomes by losing DNA and losing genes that it doesn't need anymore, and then to a mutualistic relationship where both of the organisms can lose genes, and these are usually complementary. One loses one set of genes while the other keeps those sets of genes, so they can have full complements of what they need for each of the pathways. And then finally an endosymbiotic relationship that becomes an organelle, where a lot of the genes from the endosymbiont or organelle are moved into and integrated into the main chromosome of the host cell, and so they're expressed from there. And this leads to more control of the expression of those genes from a central location, that is the main nucleus or the main genome. In the last few presentations I had shown this diagram, and it shows this relationship between free-living organisms that have large amounts of DNA and large numbers of genes, usually at least 1,500 protein encoding genes, and at least about a 1 to 1.2 megabase genome, going down into the endosymbionts and parasites, which tend to lose their genes because they don't need them anymore, so they mutate little by little and eventually disappear. and then going down to organelles, where there is more and more of the DNA lost and more genes lost. The exception is in plants, but that's a special case. One of the characteristics of an endosymbiotic event is that you get mixing of two genomes from different parts of the tree of life, the overall phylogenetic tree. So this becomes confusing in some ways, so that on the left-hand side you have a tree which would be drawn in the traditional way with the three groups of organisms, bacteria, eukarya, and archaea, coming from a last common ancestor. And then you have the mitochondrion coming over, and later a chloroplast coming over to make the eukaryotes. But in reality, what has happened is that a bacterium and an archaea came together in an endosymbiotic event to form the eukaryotes. And later, an alpha proteobacterium entered by an endosymbiotic event to become a mitochondrion, and a cyanobacterium entered to become a chloroplast in another line. And so this represents a more realistic tree. Here is a tree of life, or overall phylogenetic tree, showing the bacteria essentially taking up about 2/3 of the right-hand side of the tree, and then the archaea and eukaryotes on the left side. And then you see from the alpha proteobacteria this organism entering an archaeal cell about 2.4 to 2.2 billion years ago, and that became a mitochondrion capable of oxidative phosphorylation, which was very important at that time, as the oxygen content of the air, the atmosphere, and the seas and the water were all increasing. So this ensured that those archaeal cells that took up the alpha proteobacterium would survive. There was also possibly an initial event from the planktomycetes, which joined with an archaeal cell to become the original eukaryotes. And the planktomycetes bring over internal membranes, including a nucleus-like structure. At about 1.9 billion years ago, then, a cyanobacterium entered a protist cell that was not an autotroph, not a photoautotroph, and that cell became a photoautotroph by taking up the cyanobacterium, and that eventually evolved into the plastids that we see today. Here's a general picture of these major endosymbiotic events that led to the eukaryotes. First, you have an archaeal cell at the bottom, joining together with a planktomycete, which brought in internal organelles, internal membranes, including a nucleus-like structure. Next, into that organism, as the oxygen levels started to rise, proteobacteria could detoxify and utilize that by using oxidative phosphorylation. That was brought into the cell to become the mitochondrion. And then, as I said, about 1.9 billion years ago, inside of one of those protist cells that was a eukaryote with a mitochondrion, then a cyanobacterium entered to become the plastids or chloroplasts. So this shows how this process likely occurred. About 1.9 billion years ago, you had a cyanobacterium probably living next to a protist cell. So they probably already had formed a mutualistic relationship where they were helping each other, essentially. The cyanobacterium would have had somewhere around 2.5 million base pairs of DNA and 1,500 genes that it could bring into this cell. At some point in time, it became enveloped by the protist cell and started losing genes and started losing DNA. And this is because it was no longer necessary to retain genes that allowed it to be living outside in a different environment. And so those genes essentially, one by one, would mutate. If they were unnecessary, they would continue to mutate so that they were non-functional and then eventually disappear. Some of them, however, would move into the nucleus. In fact, we know a large number migrated and into the nucleus and integrated into the chromosomes. So copies of all 1,500 genes, or at least most of the 1,500, moved into the host chromosome. And by the time you had a plastid, only 170 genes remained out of the 1,500, and the genome was reduced from about 2.5 million base pairs to about 150,000 base pairs. When the genome of Arabidopsis thaliana was sequenced, it was found that about 4,500 genes were present in the nuclear genome of Arabidopsis, meaning that an average of 3 copies of each of the genes from Cyanobacterium had moved into the nucleus, although it turned out that some genes didn't move into the nucleus at all, and some genes were copied many, many times in the nucleus, so they exist as multiple copies as well as pseudogenes. So somewhat surprisingly, 19% of the nuclear genome of Arabidopsis it consists of genes that move there from the cyanobacterial endosymbiont. Subsequently, then, the products from that nuclear genome, products from the host genes, then moved out. The RNAs moved out into the cytosol, were translated on cytosolic or ER ribosomes, and then those proteins wound up functioning in the cytosol or in the membranes of the host organism. Also, products from the cyanobacterial origin those RNAs moved out into this cytosol, and those were translated on cytosolic ribosomes and then were imported into the plastid for their function. And if that were all that happened, then things would be fairly simple, but it was discovered that many of the products from the host genes, again, were the RNAs moved out into the cytosol, were translated on cytosolic ribosomes, but they moved into the plastid for their function. And conversely, some of the products that are of cyanobacterial origin were then, the RNAs were moved out into the cytosol. They were translated on cytosolic ribosomes and wound up functioning out in the cytosol. So now you have a combination of the genomes, but now you have combinations of where those proteins wind up, and this leads to chimeric pathways. So here is an example of that in a spinach chloroplast, and then there's the spinach cytosol on the right-hand side. And this is showing the enzymes involved in the Calvin cycle and also in glycolysis and gluconeogenesis. All of the enzymes that are colored in green originated from cyanobacteria. And all of the enzymes that are in light blue are from mitochondria. And then the ones in darker blue are of unknown origin, but probably from the original protist cell. So you can see even in the Calvin cycle, while many of the enzymes are of cyanobacterial origin, a number of them are also from of mitochondrial origin, that is, from the alpha proteobacterium. And a number of them also are from the original host cell genome, encoded from the original host cell genome. But then you look out into the glycolysis pathway and you see that some of the proteins out there are also from the cyanobacterial version. Some are from mitochondria and some are from the original host. So when you get the mixing of these genomes, then in an evolutionary sense, you get selecting out or sorting out of the various proteins and enzymes in each one of the pathways, and the one that works best for survival of that organism at that time, those are the ones that are chosen, and the other versions are not expressed, and a lot of them actually disappear and go extinct. So, for example, in glycolysis, then you have the phosphoglucokinase, the PGK up there, that's of cyanobacterial origin. And you see in the chloroplast itself, you also have the same PGK, phosphoglucokinase, functioning in the Calvin cycle. When you trace the lineages, then you can observe how these different versions of each one of the enzymes was inherited, and then how some went extinct while others were used. So if you just look at the PGK, the phosphoglucokinase, on one side, the red line goes to the archaea, and then that other line from the archaea goes up into the eukaryotes, where you get an alpha proteobacterium that comes from that left side, the bacterial side, joining with an archaeal cell, and then you have the phosphoglucokinase that's used in animals and fungi, actually came from the alpha proteobacterium. The archaeal version went extinct. Then later, the cyanobacterium entered a protist cell that already had a mitochondrion, and that phosphoglucokinase now that's used came from the cyanobacterium, and the one that came from the alpha proteobacterium, that is the one that came from the mitochondrion, went extinct. That one was not used. So obviously during evolution, one version worked better than the other for the survival of those ancestors that led to the modern organisms that we see today, and the modern combination of these genes and proteins in each one of the pathways. Then you have all of these other endosymbiotic events between red algal and green algal cells, that is plant cells, joining with other protists to become the stromatophiles, alveolates, rhizaria, excavata, etc. So all of these have then picked up photosynthetic eukaryotes as their organelles. And in a few cases, it seems that a few groups of organisms have gained not only a red algal endosymbiont, but also a green algal endosymbiont. So they've obtained a couple through these endosymbiotic events. It's beyond the scope of this class to go into the details of that, but you can imagine now that you have seen what happens just with mitochondria and chloroplasts, that these other organisms, a eukaryote into a eukaryote, are also carrying a large number of genes from each one of those organelles, and then those are moving around mainly to the main nucleus, and you get even more diversity in the chimeric pathways. So here's a diagram I've shown before where initially you have endosymbiotic events between two gram-positive bacteria to become the gram-negative, and then you have the initiation of a eukaryote from an archaeal cell and probably planktomycetes, a planktomycete, and then a mutualistic association and an endosymbiotic event between an alpha proteobacterium and a eukaryotic cell to become a mitochondrial eukaryote. Those then diversify into the animals and fungi and protists, and another endosymbiotic event between a cyanobacterium and a mitochondrial eukaryote to become the photosynthetic eukaryotes, including glaucophytes, red algae, and green algae. And all of these are carrying genomes in each one of those compartments. A second event to become pollinella, and then many more events to initiate the excavata, that are a green alga that becomes the endosymbiont of a protist, and the chlorarachnophyta, which are similar events. And then these other ones over here, the cryptomonads, haptophytes, heteroconts, ciliates, dinoflagellates, that is the stromatophiles, the alveolates, rhizaria, haptophytes. Those then take in red algal cells as their endosymbionts, and then there are events where those each take part in further endosymbiotic events. And so all of these genomes, again, are mixing. They're then sorting out as far as the genes, the duplicate genes that need to be sorted out in each compartment. And you wind up with some very complex genomes and very confusing genomes sometimes. Here's a simpler way to view that. So in the bacteria and archaea, there have been DNA exchanges back and forth, and these are sometimes endosymbiotic events, but more generally horizontal gene transfers, genes moving from one organism to another. Sometimes viruses are involved in transferring those sequences. And then you have bacteria entering an animal cell, the alpha proteobacterial cell, to become the mitochondrion, and the animal cell originally coming from an archaeal cell, joining possibly with a planktomycete to become the nucleus, or at least to bring the membranes in to form a nucleus. And then cyanobacterium entering a cell to become a plant cell, and that's the chloroplast now. Genes are moving primarily into the main nucleus. And then over in the stromatophiles, alveolates, etc., you have a eukaryotic cell that contains a nucleus, mitochondrion, and chloroplast, moving into a protist cell to become a member of those, including dinoflagellates, but not limited to dinoflagellates. And then sometimes you have that cell taking up either another eukaryotic cell or itself becoming engulfed by another eukaryotic cell. And all of those genes, most of the time, at least what I've seen, those sequences tend to move primarily to the main nucleus of that cell. And some of the organelles, those organelles within those endosymbionts go extinct, while most of the time they retain the photosynthetic member. Not always, though. In bacteria, then, they mainly pick up foreign pieces of DNA and incorporate those into their genomes. So there's horizontal gene transfer that way. Whereas in eukaryotes, they do horizontal gene transfer, but there's also a lot of endosymbiotic events that occur that cause very large horizontal gene transfers, that is, whole genomes moving from one cell into another. So this is indicative of bacteria here, where you have vertical inheritance only, and this is for 181 genomes, and this is a ribosomal sequence used in this case just to get the basic structure of the phylogenetic tree of all these bacteria. Using bioinformatic techniques, you can then sort out what would be ancient lateral gene transfers among the different lines of these along this phylogram. So now you can see there are exchanges across the phylogram of DNA sequences. You can also pick out more recent events. So these would be events that are among more closely related taxa. And if you focus just on the crown group, that is very recently separated organisms, you find there are transfers among those also. So there are transfers happening a long time ago, transfers happening not so long ago, and then transfers happening recently. When you put those all together, then it looks like this. So then the phylogenetic tree is more of a network than a simple tree. You have a basic structure for genes that aren't transferred very often. For instance, the ribosomal RNA genes are not transferred at all, from what I know. So you can use those as a basis and then look at how different genes and gene sets have moved around over time within the different lineages and among the different lineages of the bacteria and archaea and eukaryotes. If you build a network, a phylogenetic network of that, and this is for 298 genomes, then you can see that evolution is really what's called reticulate evolution. That is, you have a basic structure of an evolutionary tree, but with all of the exchanges, there are a lot of other interactions going on during evolution among those genomes. So here are the bacteria here, and then the archaea have done the same thing, and then the eukaryotes would be coming off of one of these lines from the archaea, and they add on not only the simple horizontal gene transfers, but also all of those endosymbiotic events that lead to large horizontal gene transfers. Okay, next I want to talk about eukaryotic genomes, and again, these are mixed genomes because there have been a lot of endosymbiotic events going on. So they're more complex, they're generally larger, and so I'll talk in a more general sense about these genomes. So here are some of the genome sizes of eukaryotes on top. You can see the protozoans on the bottom, the protists, and you can see it's a broad range, and this is because that includes all the way from excavata and the rhizaria, acrobians, alveolata, stromatophiles, etc. So it's a wide range of organisms. Plus, these ones, remember, have picked up, many of them at least, have picked up a eukaryotic organelle within them. So they have larger genomes, having included at least two major eukaryotic genomes. And then up into the plants, quite a broad range in those. Again, the black indicates those that are free-living. The grayish color indicates a mixture of free-living and parasites, and then the white are parasites, obligate parasites, etc. So plants, you can see going from the algae, mosses, lycophytes, monilophytes, gymnosperms, and angiosperms there, and then up into the Opisthokonta, which includes the fungi and the animals, going from invertebrates up to mammals. Also note in this one that I've included dinosaurs. The way you get dinosaur genome sizes is in some fossils you can see cellular structure, including visualization of the nuclei. And a nucleus volume is proportional to the genome size. And so this group of researchers then determined sizes for stegosaurs and other dinosaurs. And interestingly enough, they match up pretty well with reptiles and also with birds. And birds are one of the sister groups to dinosaurs that branched off a couple of 100 million years ago. So here on this tree are the eukaryotes, including kinetoplastid diplomonads. Those are the excavata, then into the chromalveolates, which are the stromatophiles, the violets, and other protists, plants, amoebae, fungi, and animals. And fungi and animals are within the Opisthokonta. So here we focus in a little bit, so I'll talk about a couple of different genomes within this group. So the first one I'll talk about is Arabidopsis thaliana, a small little plant within the cruciferae. And then another group of plants I'll talk about, and then I'll speak on the fungal Saccharomyces cerevisiae genome, as well as a couple of animal genomes. Arabidopsis thaliana was the first plant that was fully sequenced, and that's because it has a relatively small genome. This plant it has been studied for many decades now, and that's primarily because it's small, has a very fast life cycle, and many mutants have been produced, so that it's been studied, dissected essentially developmentally in many different ways. And we talked about this one a little bit before when we talked about development in plants. So here's a pie chart of the Arabidopsis thaliana genome. It has 157 million base pairs of DNA in its genome. Now that compares with about 12 million bases for Saccharomyces cerevisiae and about 3.2 or so billion base pairs in humans. It has between 27,500 and 31,000 protein encoding genes, and is able to produce about 35,500 different proteins. So there is some alternative splicing going on in Arabidopsis, but it's a much smaller amount of alternative splicing than you see in animals, for instance. Because its genome is so much larger than things like Saccharomyces, then the percent of the genome set aside for RNA and DNA metabolism, signal transduction, cell organization, transcription, etc., appear to be smaller, but it actually has more genes involved in those functions than in Saccharomyces cerevisiae. But it has quite a few more genes involved in developmental processes, since it is a relatively large multicellular organism. And a lot of its genome is devoted to responses to stress and response to external stimuli. I mean, plants are out in the sun, they're out in the cold, they're out in the heat, they're out when it's wet, when it's dry. So they have to respond to a large number of environmental conditions. So they have to be able to switch on and off different genes to respond to those. They're also attacked by pathogens constantly, so they have to react to those pathogens as well. So they have response mechanisms either to avoid pathogens or to wall off certain cells that are being invaded. Here's a look at what is on each one of the chromosomes. So you can see that the genes are pretty much spread among the 5 chromosomes, although the largest one is chromosome number 1 with a length of 30 megabases, which is about almost 3 times as large as the entire Saccharomyces cerevisiae genome. And then there are the number of genes and the number of proteins made. So you can see the number of proteins made is larger than the number of genes, which indicates that there is alternative splicing going on, so you can get more than one protein formed off of a single transcript. Also, there are 570 to 750 ribosomal RNA genes, and we discussed earlier in the semester why you have so many ribosomal RNA genes in plants, and then they're quite variable. So even in a single plant, you can have from 570 to 750 on a chromosome, and that you get recombination occurring, and it's unequal, and so one chromosome gets more copies, one gets fewer. Probably the minimum number of genes that an individual cell needs would probably be in the range of 200 to 300 ribosomal RNA genes. Then there are 689 tRNA genes, at least 70 SNO RNA genes, that is small nucleolus organizer genes, RNA genes, and then 177 microRNA genes. And then these then control both the processing of ribosomal RNA genes for SNO RNAs and control mRNA expression in the case of microRNAs. So these would be in very low number in other types of organisms. But by the time you get to multicellular organisms, plants, and animals, then these small RNAs really increase greatly in number. So as you know, when there are many different locations in the cell and many membrane-bound organelles within a cell, you can have proteins winding up in different locations. So this pie chart shows the destinations for those proteins. So about 8, almost 9% go to chloroplasts, and 3.4 to go to plastids in general, including amyloplasts and others. Then a number go to the nucleus, to the mitochondria, to the plasma membrane, other membranes, and then to various other locations such as the cytosol, ribosomes, etc. So these are where those proteins wind up in the cell. So although in bacteria, and especially in planktomycetes, you do have signal peptides that take proteins to certain locations in a cell. In the case of higher organisms such as plants and animals and protists, where you have many different cellular locations and many different membranes, there are much larger numbers of signal peptides that aid in getting those proteins to the right location in the cell. Okay, so that's the end of the first part of eukaryotes, and then next time the next presentation will be the conclusion of the class and the conclusion of the talking about eukaryotic genomes. Okay, talk to you next time. Thanks.